Members of the Senate, members of the House, ladies and gentlemen, we are here today for the purpose of signing the Cancer Act of 1971.
The National Cancer Act officially started what we commonly refer to as the war against cancer. Since the signing of the bill, billions of dollars have been used to fund a total commitment towards the conquest of cancer, with the stated goal of finding a cure.
And I hope that in the years ahead, that we may look back on this day and this action as being the most significant action taken during this administration.
Government spending has been tremendously beneficial, providing funding for basic cancer research, which is the foundation for new cancer treatments. As a result of ongoing research, we now have an incredible wealth of information, a better understanding of cancer biology, and many novel treatments. But there is still a lot of work ahead of us.
Because when we consider what cancer does each year in the United States, we find that more people each year die of cancer in the United States than all the Americans who lost their lives in World War II.
Although considerable progress has been made in the treatment of some cancers, a definite cure for cancer has not yet been found.
One of the major changes that's happened in the area of cancer therapy in the last 10 to 15 years is that we've become much better at finding a way to target cancer's fragile strengths.
Targeting a cancer's fragile strength is a key strategy. The question remains, how did our drive to find a cure for cancer start? To find the answer, we've interviewed various experts. During the annual meeting of the American Association for Cancer Research, held April 16th through the 20th, 2016 in New Orleans, we sat down with Dr. Amit Kumar, an expert in cancer diagnostics, to talk about how to change the war on cancer. Dr. Kumar is the Vice Chairman of IDIS Corporation and Executive Chairman of Anixa Diagnostics Corporation, which is developing a blood test called C-Check for the early detection of cancers.
In the 1960s, cancer was really not a focus of the medical community. In 1971, when President Nixon signed the National Cancer Act, that brought presidential focus and national focus to this area. And since that time, we've done a tremendous amount of work in understanding the basic physiology and biology of cancer. Since that time, we've been focused primarily on curing cancer because human beings are reactive in nature, and when someone gets sick, we want to try and cure them. However, we've learned since that time that cancer is comprised of several hundred diseases, and trying to cure cancer as a broad general category is a very, very difficult task. We've made tremendous progress with chemotherapies and radiation, as well as recently with antibody-drug conjugates and immunotherapies. But in general, chemotherapies, for example, kill cancer cells only a little bit faster than they kill normal cells. So it's really not a fantastic therapy. Some of the new therapies, however, are making tremendous progress, but they only work on a small subset of patients. And again, the focus has been on trying to cure people who have got advanced stage cancers, primarily metastatic diseases.
This predicament has made many scientists wonder if it's time to rethink our approach in fighting the war on cancer. Do we need a new strategy, or is it too late? And have we altogether failed at fighting this war?
In the last 45 years, I think we made a tremendous amount of progress. We've really not failed, that's probably too strong a word, but I think we've learned that the battle to cure cancer is not as easy as we had initially thought it might have been. It's not like a single infectious disease, for example. And as a result, we feel that there may be some new paradigms in how we look at cancer. If you look at the history of treating cancer, most patients, when they're caught early, tend to live and have better outcomes, regardless of how one treats them. Patients who are caught at later stages, primarily stage 3 or stage 4, which is usually metastatic disease, don't often have good outcomes, and many of them die. It's very, very difficult to cure them.
Today, our response to cancer largely remains reactive. Cancer statistics show that the medical community's focus on curing advanced stage cancers has, in some respects, been counterproductive in reducing cancer mortality rates. As a result of late-stage diagnosis of cancer, treatment options may be limited and prognosis continues to be gloomy. While cancer drugs and therapies are important, a greater impact on reducing cancer deaths and improving quality of life for cancer patients can be achieved by early cancer detection and early intervention. Early screening, detection, and prevention have had a tremendous impact in cardiovascular medicine. The standard treatment of heart disease was to wait until the patient exhibited signs of the disease, much like our current approach to cancer. But over the past years, our approach to heart disease dramatically changed to one of prevention and early detection. The result has been a 60% decrease in heart disease-related deaths since 1971. Emphasis on early cancer detection is expected to yield similar results. This need for early diagnosis and early intervention emerges if an improvement in patient survival is desired, and it will change the survival advantage patients may have.
Statistics clearly show that early stage diagnosis results in a much better outcome, greater chance for continued life. 5-year survivability is a statistic that we use in the industry. So any time that you can catch malignancy early, and the earlier the better the outcome. It's been difficult to diagnose patients early because the measurement techniques and the analytical techniques that we've had in the past have not been able to identify the specific markers that are indicative of an early stage disease. However, things are changing. We're making tremendous progress in technology, in computation capability, and the ability to identify specific biomarkers that are indicative of pathologies in the body.
Novel diagnostic tools are being developed to aid early diagnosis. This relatively new field of identifying biomarkers for purposes of early cancer detection is something referred to as liquid biopsies. We are now at the forefront of a new wave of early cancer detection technologies that has been fueled by innovative startup companies in the private sector.
The liquid biopsy concept is a relatively new concept that's been developed over the last few years. The basic premise is that blood bathes the whole body and carries within it markers that are indicative of the health of all organ systems. And traditionally, we've always used blood chemistries and blood analysis to identify other issues within the body, within the patient. And now we're able to look at specific markers that are indicative of cancers. In fact, new biomarkers are being discovered every day. And the liquid biopsy industry, if I can call it that, is growing very rapidly. Lots of companies are working in this space. Many academics are working in this space. And I think it's going to revolutionize the way we analyze cancer and evaluate patients.
Liquid biopsies rely on blood samples drawn from a cancer patient to analyze trace amounts of free-floating tumor DNA and other biomarkers found in the blood. Other bodily fluids, including plasma, urine, saliva, and even breath, are currently being studied for early indicators of cancers. Liquid biopsies may actually provide a more accurate picture of cancer, as genetic sequencing of free-floating tumor DNA may better capture the diversity of genetic alterations found in cancer cells in different parts of the body, including the primary tumor and metastases. As a minimally invasive test, liquid biopsies offer several advantages over conventional tumor biopsies. Since surgery is not needed, patients can be tested more frequently.
There are a couple of points to focus on with respect to liquid biopsy. Liquid biopsies are often used today for the purpose of monitoring therapy for patients. Because if someone has a particular type of cancer, it's not easy to get tissue every time. You want to monitor how that cancer is doing. You can certainly do some imaging, but you don't want to cut into the patient every time you need to evaluate how the therapy is doing. So if you can look at specific markers that are circulating in the blood and watch their behavior over time, you can monitor how the patient is doing. And companies have developed tests to do that. Many of those tests have been commercialized under a 1988 law that was passed called CLIA, referred to in the industry as CLIA. And the US FDA is evaluating whether they want to regulate those tests, and we'll see what happens. That's still up in the air.
Testing for mutations in the blood can help identify patients who may benefit most from certain drugs or drug combinations. Liquid biopsies are expected to become standard practice for testing new drugs and monitoring response to current therapies. Offering a more accurate, minimally invasive tool may change the patient's compliance, their acceptance, and experience.
Patient compliance is much better if one has to simply take a blood sample as opposed to putting a needle into the patient or cutting into the patient. So from the patient standpoint, it's a tremendous revolutionary advance. For the physician, it's a good way to monitor how that patient is doing. But liquid biopsies are still relatively new. Not everyone uses them. The quality of performance of these tests is still improving. And the biomarkers that we're looking for in liquid biopsies are still being discovered. And there are very, very many more that need to be discovered, validated, and clinically utilized.
We are at an inflection point in the war against cancers, with potential changes on the way from what some might consider to be disruptive technologies. Change is long overdue. The sooner that we begin to chip away at the monolithic thinking of the past decades and open our minds to new approaches to how we think about the war against cancer, such as early cancer detection, the faster changes will happen and the better our chances of success will be. Anixa Diagnostics is developing a platform called C-Check for inexpensive, non-invasive cancer screening blood tests for the early detection of tumor-based cancers.
C-Check is a test that is, we feel will be able to look for multiple types of cancer. We've started working with breast cancer. We're expanding into other types of cancer, but we've done a very small number of patients right now. We're evaluating how possible this technology will be to identify early stage. We don't know how early stage yet, but we certainly see the indicators of cancer in stage 1 and 2 breast cancer, which is the situation where you can treat the patients effectively. As we go forward, we will continue evaluating other cancers. We will do larger numbers of tests. We will continue doing blinded studies, and then eventually submit this technology for FDA approval. And we'll choose one or more types of cancer or an adjunctive test as a initial commercial launch.
One of the key issues in the development of novel technology, such as C-Check, is the requirement of regulatory approval.
In general, there are two possible ways for companies to launch new tests. One is under the guideline known as CLIA, which was a law passed in 1988. And in that procedure, one has to develop a test, validate it in a laboratory, and provide it as a service to customers, which are physicians ordering the test for their patients. Another approach is the traditional FDA approval route, which is either a 510K or a PMA, pre-market approval submission. And there's a little bit of controversy in the industry right now. The FDA feels that they may want to regulate CLIA tests. Many companies in the industry are pushing against that. Our approach, we've decided, is to go down the FDA route for a number of reasons. One is we want to be able to proliferate our technology and our test as widely as possible, and we feel that the best way strategically is to get FDA approval for that test.
In the landscape of diagnostic tests, including companion and complementary diagnostics, C-Check fills a unique unmet medical need. But one concern in the development of diagnostic tools is the issue of false positives or false negatives, and the impact this has on patients' lives. A false negative means that a patient with a disease is misclassified as not having the disease. In contrast, a false positive means that a patient without the disease is misclassified as having the disease. The consequences of both false positives and false negatives need to be carefully considered in assessing the usefulness of diagnostic tools.
There are a lot of revolutionary changes occurring in diagnostics, in companion diagnostics, complementary tests, as you noted, evaluating what's the best way to treat a patient, evaluating whether a particular drug will work on a patient or not. There are technologies that are, as I noted, monitoring how a patient is doing on therapy. So there are lots of things that are happening. Our focus is on early detection. Now, early detection has a fair amount of controversy associated with it, early detection or screening. And the reason is that while early detection can identify patients who need therapy, who have cancers at an early stage, often you also create what's known in the industry as false positives. Patients that may not have the disease or do not need a particular treatment, and yet they end up being anxious because they were a false positive, and sometimes they get overtreated. A classic case is the PSA test, which has been used for years and is still used millions of times annually in the United States. And many decisions are made based on the PSA test, and it turns out that a lot of those patients that get prostatectomies or even just get biopsies didn't necessarily need the biopsy, because the PSA test, while it is able to identify patients who have aggressive prostate cancer, it also, for every one patient that it identifies that for whom their life is saved, there may be 10 other patients that end up getting overtreated and overdiagnosed and overtreated and have medical procedures that may not be necessary, that are painful, and that are costly. So that's the controversy. The key to any type of new early detection technology has to be, one of the keys is that it has to have very, very low rates of false positives. When you think about screening, you have to make sure that your test has as few false positives as possible. And let's just, for the purpose of explaining how that works, let's just use a little bit of a quantitative analysis using round numbers. Typically, if you test 1 million patients, about 5,000 of them might have cancer. And if you have a test that is 90% accurate in calling those patients who have cancer, something we refer to as sensitivity in the industry, then that means of those 5,000 of the million that do have cancer, 90% of them will be called correctly. So that means 4,500 patients will be called correctly as true positives. We'll miss 500, but hopefully those 500 will be tested again next year. We'll catch most of them next year, and so forth. Now, the big issue, though, is of the 995,000 of those million patients that don't have cancer, if your test is 90% accurate in calling them correctly, that means 10% of those patients, which is 49,000 patients, will be called false positives. And that means those 49,000 patients will have a tremendous amount of anxiety. Many of them will go and have additional procedures requested, done, or they'll request from their physicians, which include additional imaging or perhaps even biopsies. And that's a big number. And so that is the main reason why people are concerned about screening. Because to save those 4,500 patients' lives, you're creating almost 50,000 patients that have a tremendous amount of anxiety and are probably going to get overtreated. So the key is that any screening test, whether it's for asymptomatic patients or recurrence testing or even adjunctive testing, has to have a very, very low rate of false positives. So patient quality of life is critical. We know that when patients have stage 4 cancer, the quality of life is extremely poor. They're in a tremendous amount of pain and discomfort. They have gone through numerous procedures that have probably weakened them. Today, many of the therapies that are used will provide them with a short additional lifespan of a few months to a couple of years maybe. There are obviously anecdotal evidence that there are some patients that go into complete remission, but that's rare. However, with some of the new therapies, that is happening more and more often, yet still late-stage patients are having a very, very poor quality of life. In addition, something that is perhaps not spoken about as much is that these late-stage patients, even if they have the best healthcare coverage and insurance, tend to have to deal with tremendous financial burden in treatment, especially because most coverages will cover a certain portion, but not a lot. And treatment at that late stage of life is very, very expensive.
While there are many diagnostic tools on the market, new tests are expected to improve the health-related quality of life of many cancer patients. But to be truly successful, adherence and acceptance of invasive and uncomfortable diagnostic tools is still required. Based on a simple blood draw, new tests may be less invasive.
The goal of the C-check is to try and enable a very highly compliant test, which is, when I say highly compliant, I mean it's something that's very easy for patients to do. It's simple blood draw. One thing I want to note is that there are good screening technologies out there like colonoscopy, but people tend not to do them as often as they should because of the discomfort and issues associated with an invasive procedure like that. We want C-check to be a very simple procedure, a very simple blood draw for the patient. And our goal is to, from a performance standpoint, is to have as low a rate of false positive as possible. And we think that that's going to be possible, at least based on our initial testing in the laboratory, but we have to perform larger numbers of analyses before we can confirm that.
The concept of screening asymptomatic patients for early signs of cancer is as much a departure for the patient as it is for the existing cancer establishment. No presumably healthy individual wants to think about the possibility of getting cancer, or the concept of regular cancer screenings. This is why the national dialogue about cancer needs to change. If we can identify cancer through early screening, as opposed to finding it in a late stage, the effectiveness of novel diagnostic tools may be high enough to replace existing treatments like surgery, radiation, and chemotherapy, and altogether improve outcomes. This may lead the way to a bright new future of hope and healing. Next, in Changing Strategies in the War on Cancer, Part 2, the complexities in manufacturing highly potent anti-cancer medicines.